My name is Mauro Baronpriori. I'm an environmental engineer from NYMPHE, and it is a real pleasure for me to welcome you all to this stakeholder technical workshop of the European NYMPHE project. I would like to extend my greetings both to the participants here in Ferrara and to those joining us remotely. Let me say that it is great to see that we have people registered from all 5 continents today. So, benvenuti. For those who may not be familiar with us, NYMPHE, which is the organizer of this event, is Eni's environmental company. We are dedicated to the remediation of contaminated sites, the treatment of water and waste, and the promotion of circular economy initiatives to restore and give new life to contaminated areas. Within NYMPHE, I work in the Research and Innovation unit. Our mission is to study, develop, test new solutions for soil remediation and resource recovery. Research and innovation are central to our work. They allow us to address complex environmental challenges, explore emerging technologies, and collaborate with partners across Europe. This is exactly the connection with the NYMPHE project. Through NYMPHE, we have the opportunity to work side by side with universities, research centers, and companies from different countries to test advanced bioremediation approaches. Here in Ferrara, we are running one of the project's pilot tests, applying microbial-based solutions to remediate groundwater and soils, both contaminated with mixed pollutants. Today's workshop is an important interactive moment for NYMPHE. Our goal is not only to share the progress and the results of the research, but also to receive valuable feedback from stakeholders, scientists, regulators, industry representatives. So your feedback on the technology we are developing. We want to better understand the potentials, but also the barriers and the real-world challenges for future applications. This event is organized into 3 technical sessions, followed by an open discussion. The first session will focus on assemblies of biologics and technologies for wastewater treatment. After a coffee break, the second session will present the assemblies of biologics and technologies for soil bioremediation. The last session will explore innovative approaches for the performance improvement of microbes and microbiomes in bioremediation. We will finally close with a roundtable discussion to collect the augmented perspective. As participants, in fact, you bring an important perspective, as Elena Biagi will explain shortly. NYMPHE is not only a research project, it is a platform to test and validate solutions that can be applied in the field. Your feedback on feasibility, regulatory aspects, costs, social acceptance is key to guide our work in the right direction. I hope you will find today's workshop both stimulating and inspiring. Please take this opportunity to exchange ideas, ask questions, and share your views. Your contributions can help shape the development and future application of these technologies. Before passing the floor to Giulio Zanaroli, coordinator of the NYMPHE project, who will explain it in more detail, let me take a moment to thank all the NYMPHE partners, especially University of Bologna and Innovo, and all the Remtech Europe staff for their contribution to the organization of this workshop. Thank you for your attention.
Thank you, Mauro, and welcome to everyone here in the room and online also from my side. I am Giulio Zanaroli from the University of Bologna, and I am the coordinator of the project NYMPHE. Today, we will introduce you to the project, and then we will show you only part, of course, of the activities and technologies that we are developing within and testing within our
our project. So I will move directly to the presentation, the introduction to NYMPHE. NYMPHE is moving. No, it's not. Okay, sorry. So NYMPHE is a research and innovation action that has been funded within the Horizon Zero Pollution 110 topic, started on January 2023 and will end by the end of 2026. So 4 years project. We received about 5 million, 0.5 million euros funding by the European Commission. It consists of a quite large consortium of partners, including universities and research centers, but also small and medium enterprises, a large company, ENI Wines, and a couple of NGOs, which are collaborating from 12 different countries. The main objective of the NYMPHE project is to develop bioremediation, but also revitalization strategies for contaminated sites, exploiting bioremediation systems that consist of multiple biologics, and I will explain a bit more in the few minutes about this, this approach. In particular, we wanted to assess them from multiple perspectives, so not only the technical, but also the environmental, the economic, and societal ones, and of course to showcase them for real contaminated sites. The overall ambition, of course, of our approaches and technologies is to remove the majority, so at least 90% of the main pollutants present at the target sites. Here you see the sites targeted in the NYMPHE project to develop our technologies. We have an industrial site contaminated with soil and groundwater contaminated by complex mix of hydrocarbons. We have an agricultural site with soil contaminated with microplastic and pesticides. Then we are working on wastewater treatment technology, in particular municipal wastewater treatment technologies, because we, of course, municipal wastewater is a source of contamination for mainly surface water and then soils and groundwater. Here we target pharmaceuticals, complex mixture of pharmaceuticals, including antibiotics and microplastics. And then the fourth contaminated site is a river in Germany, where we address the so-called hyporheic zone, so surface water and sediments that are contaminated by, again, complex mixture of pharmaceuticals and trace organic chemicals. We also have in the project a fifth site, which is not a contaminated site, but it is a special site where we are going to test our remediation technologies, exploiting genomically edited microorganisms that cannot be, for the moment at least, tested in the open environment. Here you see highlighted with the red box the two sites that the technology I will be, or we will be, sorry, showing you today are targeting. The NYMPHE concept tries to address the high complexity of real contaminated sites, where bioremediation can be limited by a number of factors. We may include the type of pollution, in particular highly complex pollutant mixtures, but can be also limited by environmental factors and an important aspect by lack or low performance organisms acting the remediation process. And the approach or the concept we want to exploit is based on, let's say, inspired by the complexity of these systems. So it is considering the whole system, including microorganisms, matrix, environmental factors, and so on. And in particular, it is based on the use of associations, of multiple biologics, on the development of a toolbox to improve the biodegradation efficiency of those biologics, and also it is based on the management of microbiomes in the contaminated matrix during bioremediation, which is based on their modeling. The NYMPHE approach is consisting of three main tiers. First, we obtain and select different biologics, which may include enzymes, different microorganisms, plants, and animals that are suitable for targeting the main pollutants occurring at the contaminated sites we are considering. And then we assemble those different biologics in a rational way and test them, introduce them in the real contaminated matrix from the site at the laboratory. In this case, we do not look only at the degradation efficiency, but we also collect information on how the microbiome in the system is changing and behaving, and this is an important input for the modeling of those microbiomes, because we aim to identify keynote species and metabolic activities that are need to be boosted to increase the bioremediation efficiency of the system. At the second tier, is dedicated to improvement of the performance of those biologics, in particular the microbial ones. Here we follow different approaches, so both looking at the system level. So, for example, developing model-based microbiome modulation strategies, identifying some amendments that are able to shape and boost the activity of the microbiome. But also in the project, we are developing tools to improve microbial communities or single microorganisms without genetically modifying them, so natural evolution or introducing genomic variability in a microbial community. And finally, we also adopt some engineering, including, of course, some genetic modification approaches. Here again, you see highlighted in the red boxes only two of the approaches that will be presented today, although some of them has been or will be targeted a bit also in some technologies, for example, the natural evolution of microbial degraders in membrane bioreactors. Then the project in a third tier actually test the assemblies of biologics that have been pre-assessed in the laboratory, introduces and tests them in the real contaminated sites. So we perform field testing, also to collect, let's say, the complete information for the holistic assessment. So not only the technical, but also the environmental, economical, and societal aspects. And here I wanted to highlight that in the NYMPHE project, in order to achieve some of, or to complete some of this assessment, we are developing some innovative tools, for example, an innovative integrated evaluation tool to assess the environmental quality and the risk of the treated matrices, and also a mathematical model that is forecasting the environmental impact of bioremediation and can support in the evaluation of the revitalization status of the site in relation to the Natura 2000 ecological requirements. So concluding, what we expect to achieve by the end of the project, and of course we have already achieved partially some of these, let's say, objectives. I mean, NYMPHE wants to provide a set of systems of biologics able to address efficiently certain types of polluted sites, but also delivers microbiome management strategies to improve the performances of the bioremediation, delivers tools for further improving the performances of single biologics or microbial communities, and finally assesses, let's say, from the environmental and technological point of view, those technologies at the field scale. So we expect a number of environmental, toxicological, economic, and societal impacts that hopefully will improve the status, let's say, of these of these sites. And with this, I conclude this short presentation of the NYMPHE project, and I would like to leave the floor to Professor Biagi, who will actually explain better in a few words which is the aim of the meeting today, and the most important aspect for us, what we would like to receive also from you, or expect from you, in terms of active participation to this event.
Okay, thank you. I'm Elena Biagi. I'm from the University of Bologna, and I'm part of the coordination group of NYMPHE project. And my colleague, Professor Zanaroli, already explained you the ambitions, the high ambition of NYMPHE project, but NYMPHE doesn't want to stay in the lab, doesn't want to be confined into test sites. We want to reach outside of our world. We want to engage stakeholders and then users of the technology that we want to develop inside NYMPHE. So with this, the aim of this stakeholder meeting, which is actually the second one and was foreseen in the project, is first of all to learn how to efficiently communicate with stakeholders. This is very important because, as I said, we do not want to stay in the lab and in the test site. But most importantly, we want to collect feedback. So the communication doesn't want to be only in one direction. It needs to come back with inputs. It needs to come back with opinions and especially feedbacks that help us to direction our future research and development actions. So the stakeholder meeting today will focus on explaining only part of what we are developing in NYMPHE, what is now at a more advanced stage, but of course this is not limited. NYMPHE is not limited to what you will see today. However, we want stakeholders, we want end users to evaluate what we are doing. And we want you to help us identifying limits and challenges, especially that will be connected to the application of the technologies in the real world. We want you to help us identifying barriers that will prevent a successful exploitation. And these barriers might not be related to the technical side only, but might also be related to the acceptance of the public with respect to our technologies or financial constraints. And we will collect your opinions. So, this is, of course, the title of our meeting today. This is the agenda, and I want to attract your attention on the fact that each one of the sessions will be followed by a question and answer moment, but most importantly, by a moment in which we will collect your feedback. How do we will collect your feedback? Through a questionnaire. Our colleagues from Innovo, which are responsible for the communication of NYMPHE, has prompted some questionnaire three different questionnaires, actually different from each one of the sessions. You will be able to access this questionnaire through a QR code, and we want to collect your input. These are very important to us, very important because it is one of the main objectives of NYMPHE to have stakeholders evaluate what we are doing. And questionnaires, of course, the results of these questionnaires will help us maximizing the impact of NYMPHE. So I really invite you to use the QR code through your smartphone and answer our question, because this input will be a key point of our future actions. So thank you very much. So the next one, I guess, is Ramiro. And I leave the stage to one of our experts.
Thank you so much, Elena, and all the NYMPHE coordination team. This talk was supposed to be called by Abraham. Unfortunately, he's not here. My apologies. Day one, substituting him, he unfortunately couldn't make it, but I will try to satisfy your expectations. I'm Ramiro Blasco. I'm a research and development project manager at NYMPHE. We are SME based in Spain. We basically have our own, we develop our own technology for wastewater treatment, wastewaters from industrial sites or urban wastewater. I'll show you in this presentation. You will have a good overview of what we do. I don't know, sorry, the ones that you are here, you cannot see the whole slide, but I don't know, the one fact that is surprising for me is that we have 48 million people in Europe that is living in small towns. When I refer small towns to settlements of less than 2,000 inhabitants. Well, these people, the administrations that are running those settlements, they are obliged by the European Union to treat the water. This is going to be even more restricted by the new wastewater treatment directive, that is even decreasing the limit in which the towns they have to implement secondary treatment for treating the wastewater, and it's going to be the limit of 1,000 inhabitants. So it's going to be even more restricted. Therefore, more people will be requiring those treatments. Historically, they have been implementing these conventional technologies, which have high operational costs, low footprint, and high impact to climate change. A couple of decades ago, the EU appear in the market other alternatives like, for instance, wetlands that they also satisfy the requirements. And they are improving the facilities, giving having low operational costs, high footprint, and low impact to climate change. Still, the drawback of high footprint and what NetFilter brings to the market is a solution called Metland. This concept is breaking all the rules and is offering a solution for this kind of settlements and administrations with a solution for a technology with a low operational cost, low footprint in this case, and low impact on climate change. I will show you why it has low footprint compared to conventional wetlands or conventional technologies. This technology, this concept, Metland, was born in 2014 after a successful project called iMetland. This was an EU-funded project, so we are very proud to be born under the umbrella of the European Union. We are considered a nature-based solution, and we can be, our solution can be implemented not only in those facilities or those environments of remote small populations, but also we are present in big cities like Madrid downtown. We are in buildings. Well, it's enough versatile technology to be implemented in different situations. The main difference with wetlands, with constructed wetlands, is that the material in wetlands are inner materials, so from the microbiological point of view, it requires a physical contact between microorganisms to do their work, to treat the water. In our system that you can see on the right slide or in the right figure, what we propose is one new material. So we are also playing with the material field of the bed. This material is conductive, which what makes is all the microbiology present in the bed to be connected. So that material is in a way a highway for electrons. Therefore, it's enhancing the activity, enhancing the power of the system, of the bacteria in this case. They don't need to be together anymore. They can be very far away from each other and still doing their work. Therefore, what we achieve is 10 times more efficiency, 10 times more power of these systems, and compared to wetland, traditional wetland. With this slide, I wanted to show you some results. These are coming from a site of 2,000 equivalent inhabitants in the southeast of Spain. The place is called Otos. We've been running and operating a wastewater treatment plant based in our technology there since 2018. You can see there the baseline of discharge limit on the upper graph. You can see all the dots that are scattered on the top of the upper part of the graph are the inlet organic content, and the outlet, you can see it on the bottom line, very close to the 0 level, which we are very satisfied. And the graph that you have in the bottom, you can see that all these dots, which are the outlet, water, organic content, they are below the discharge. We are very satisfied with this system. It's our little baby in that sense because it's the first one. So we are very satisfied and still running. They are very happy with us. If you want, obviously you can visit it whenever you want. Different applications of the MENDAC technology, not only this low inhabitant population sites, but also we are present in single-family houses, treating urban wastewater, and even in industrial wastewaters that our system has proven, or our technology has proven to be very successful. For instance, fields like oil and gas, food and beverage, pharmaceutical industry. You can see there in the slide the different configurations, constructed, modular, single boxes of 1 cubic meter, so very compact for treating 4 to 5 people houses. In the context of NYMPHE, we looked on how our system can treat micropollutants in urban wastewaters. These results were the first ones, our first insights on that issue, which were under the umbrella of a project called Electra, which was at EU China flagship. You can see that the removal of pollutants between the aerobic phase and anaerobic phase, because also our system can be working in both configurations. Therefore, we have a lot together trying to decrease these micropollutants. And you can see down left part, the graph with the influence percentage. Sorry. the total concentration of emerging pollutants decrease after passing through our system. Not only decreasing the concentration of micropollutants, but we go beyond that. We regenerate and we reuse water. The water that we treat, we reuse it for agricultural purposes. You can see here vertical gardens and hydroponic columns that are growing, or the plants are growing with our reused water. And we are also attracting key species in the ecosystem, like pollinators. This is something that we are exploring now. We're very happy if you guys want to work with us in that sense, because we are very interested in that aspect of our technology as well. You can see here an overview of the NYMPHE site here under the NYMPHE project. We are currently treating around 10 cubic meters of urban wastewater per day. This is a system that is doing that job, and we are getting quality waste treated water with quality for reuse. And you can see that we are reusing it in the vertical garden there. Right now, in the years, even better after all the high temperatures and the good weather during the summer time. So, well, you can see that it's very interesting as well, not only the treatment, but also the beautification of the area of with technology. And there, another national project, this is called NYMPHE. We also play with the dimension. We are not satisfied with one system, but we also play, having blocks and playing with this 3D Lego. The concept is the same technology, a biofilter, but in this case, we have different configurations in the same site. So you can see on the right, a render, beautiful render, but the important thing is that right now it's a reality. 2 weeks ago, we set up this nice concept of 3D Lego, and each module is doing a different job. So at the end, we can also play with different configurations, and in order to get rid of the most of pollutants, which is also very linked to NYMPHE, micropollutants and different difficult, let's say, pollutants to remove. This system is located in Seville, in the south of Spain. Again, if you are interested in visiting it, I will be very glad to have you there. Moving to also the NYMPHE project, we also have other configurations that we are also trying to reduce the amount of contaminants. Here you can see on the right a video in which you can see the wastewater, the raw wastewater, and the system setup that we have. We have a primary treatment via NYMPHE tank, and then you can see there the configuration. In this case, a very compact system. It's roughly 2 square meters of surface. You have 4 elements there, 4, let's say, boxes in each box. So we have 2 boxes that are working in aerobic, let's say, aerobic treatment. And then we have another box that is collecting the water, and we are using it for storing the water. And then the 4th box is an anaerobic one. So we are playing also with anaerobiosis. These boxes that you see, apart from that one, is a control panel, but we have another box there attached to the system that is a UV light. So we are disinfecting the water in order to reuse it for agricultural purposes. You can see how it works. At the end of the day, it's a filter. We pump water on the top. It goes down. So it's kind of a shower of water, wastewater. While it's going down, the bed is being treated. Okay. We pump, depending obviously on the amount of contaminants. We have to pump 1 time or 2 times, or depending on. You can see there the quality of the water that goes out of the system. and you see the difference between the inlet and the outlet there. You can see that the filter works perfectly. In terms of organic content, we are satisfying administration because we are below the discharge limit. So, like in life, it's important who you partner with. In our system, we do know that there are communities working together. Under the NYMPHE project, what we are doing is also trying to see how they are working together. In this case, we were trying or putting together Pseudomonas tutida and Geobacter sulfur-reducens. We know very much the Geobacter because they are the ones exchanging the electrons in a very easy way. Pseudomonas tutida is a good a microbe that we know is very good at degrading some pollutants, and we just wanted to have them together working closely in our system, in our bed. The results were very nice because we could see that Pseudomonas by itself can do the job, but Geobacter by itself cannot do that. And whenever it's with Pseudomonas as a co-culture, like working together, the energy that we measure there, the amount of electrons that are floating along our bed are higher. Therefore, what we can conclude is that they are doing the job, they are degrading things together. On the other hand, Geobacter by itself couldn't. So it's a way to prove that syntrophy is a key aspect of our system, and having many different microbes working together is the key here. Also under NYMPHE project, we are working with other partners. In this case, Victor De Lorenzo's lab. We have representative of the lab here. You will see more in detail what they are doing. But in our case, they are helping us to engineer a little bit more our system, give more quality and more insights of what is happening inside our system. And they are developing a toolbox, a biological toolbox to increase the evolution adaptation of different electroactive microbial communities in order to improve bioremediation, therefore boosting the efficiency of our system. So we are very happy. We hope we keep on collaborating not only with Lorenzo, but other also partners in the NYMPHE that can improve significantly our system. Also in NYMPHE, we are working not only with microbes, but also with the material, our conductive bed. We are actually using the microbes that are developed by Victor De Lorenzo's lab and adding them to the material directly. So we are capable of adding the microbes directly to the material, which will be seen on the top of our systems, and while feeding the system, the community will be slowly colonizing the system, and therefore our efficiency is boosted. You can see here on the left how the pelletizer works. Sorry for the noise. But these pellets are already included inside the microbes that will be lately used for doing the job in the system. This case is Geobacter, and what we proved is that the concentration of microbes in our system is higher due to this extra push that we are giving to the system by adding externally more communities, so biomonitoring actually the system. And finally, I wanted to show, coming back to the beginning of the presentation, a video in which you could see the system that we have. So this is a camping site in the southeast of Spain, a wonderful place. We wanted to install our system in this camping site. It's remote, 1,000 inhabitants equivalent. They had a conventional technology there. They were very unhappy with it. It was very noisy, very smelly. The odors were difficult to handle, especially in a camping setting, which you are trying to go there just to enjoy the environment and to be relaxed with your family or alone or whatever. And so we retrofitted our technology in a conventional site. We basically fill the secondary tanks with our biologics and our material. Right now, the owners of the camping site are super happy. The place now is visited by the clients, by the customers. As you can see, it's the same, biofilter in a different scale as I showed you before. No odors, the noise of the air pumps are not present, so it's a very pleasant technology. We not only treat but also reuse the water, so they are very satisfied. It's a very successful story, and again, super happy to having you there. It's a magnificent area, southeast of Spain, the Cabo de Gata in Almería, and well, it's a fantastic place to go and also to see our technology. So thank you so much for your presence, for your interest, and well, there will be some questions later, so we can discuss a little bit further. And thank you so much for it.
So, I will present the work of Francesca De Maria. She's a PhD student in my group. So, as you know, we are all here because of pollution by micropollutant. And we know that wastewater treatment plants are not designed especially to remove the micropollutant, and pharmaceuticals is a question of very low concentration in the presence of very high CO2 content. So on the top of that, there are some fluctuations, seasonal fluctuations, for instance, for the use of the pharmaceuticals, and also variation in terms of pharmaceuticals present in the wastewater. And in addition, you have different wastewater treatment processes. And for instance, they can have different performances. In Switzerland, we have a treatment step, for instance. And on the right side, you can see some relevant pollutant and maximal detected concentration in the wastewater. So they are above tens of microgram per liter. So in the NYMPHE project, our idea was to take advantage of operating membrane bioreactor operated at infinite retention time. So meaning no removal of excess sludge. This is a way for us to keep slowly growing degraders, and thus to achieve a higher level of biodiversity. So here you have the lab scale system. They are about 1.5 liter volume. And we targeted 6 pollutants that are paracetamol, enalapril, coffee, atenolol, ibuprofen, and diclofenac. So they were operated in a continuous flow with a HRT of about 62 hours. So the work plan was as follows. We acclimatized the microorganisms, the microbiome, in our membrane bioreactor system at high concentration of pharmaceuticals. We applied them synthetic wastewater at 100 milligram per liter concentration each. And then we operated until we got stable removal performances of the MBR system. And then we switched the concentration in pharmaceuticals to 1 milligram, down to 1 milligram, to create a stress to the microbial community microbial community. And then once removal performance was stable, then we switched again to the initial concentration to see if the overall degradation performance was affected. So this was the working plan of Francesca. So here you can see the first acclimatization phase. You have the 6 lines that represent the various NPR, and in the column you have the removal of the pollutant. So this is the y-axis is indicating the percentage of the removal. And what you can see is we could operate 6 membrane bioreactor system with stable removal performance for the 5 pollutants. Only diclofenac was not degraded. Then in terms of the microbial community, we had one NPR that has, for any reason, a different microbial community evolution, while the 5 other ones were showing quite similar composition with predominantly Renarella and let me see the last one. The yellow one is Leucobacter. While in the NPR number 1, we had another major representative. So keep in mind the NPR 1 showed a different evolution composition. Then we also investigated the biodiversity, the microbial diversity in our system, because you can guess that applying a combination of 6 pollutants in comparison to a regular wastewater, you would decrease in biodiversity, and this was not the case at all. So it was also indicated that we managed to have the slowly growing microorganisms kept in the system. Now we went with the stress, and what you can see is with the low concentration, 1 milligram per liter concentration, the membrane bioreactor were all able to deal with this low amount of pharmaceuticals within a few days, and that we managed to also keep a constant removal of those pharmaceuticals at lower concentration. And then we switched back to the initial concentration of 100 milligrams per liter. And what you can also see in this series of graphs is that the microbial community in the 6 NBR recovered the degradation performance, and the NBR1 showed a slightly different stability in terms of removal performances. Then we were looking at the microbial diversity in our system, and surprisingly, after coming to the recovery phase, we ended with almost similar microbial community as during the acclimatization phase. So meaning that in between, despite the stress caused by the low concentration, the community was again similar to the first acclimatization, at the end of the first acclimatization phase. So having Leucobacter and Renarella in the 5 systems, and interestingly, the membrane bioreactor 1 was continuously evolving in a different way, where a major representative was Achromobacter. And with this series of experiments, we were still working with very high concentration of pharmaceuticals. So the next step was to take inoculum from NPR6 that showed interesting removal performances, and we incubated in batch mode the synthetic wastewater that was spiked with 5 of the pharmaceuticals assessed at a concentration of only 20 micrograms per liter, which is relevant for any environmental concentration of the pharmaceuticals. And we could show that within 3 days, all the 5 compounds were entirely degraded and compared to, of course, abiotic controls. So the next step was to test our system, our idea, and we wanted to test the efficacy of the removal of those pollutants from our NBR, so taking the microbial community from our NBR, combining them together from NBR 2 to 6, because I told you NBR 1 evolved in a different way. And the idea was to apply those microbial communities as a polishing treatment step of biologically treated wastewater. So wastewater where the COD content is quite low. And this work was done by Francesca in collaboration with our NetFilter colleagues, especially Ramiro. So we worked there with the effluent of the NetFilter treated wastewater. And we applied this wastewater, or this wastewater was fed to a membrane bioreactor that was upscaled, containing 7 liter. So it's a 7-liter system with aeration. And then withdraw the permeate using a peristaltic pump. So the system was operated at hydraulic retention time of 33 hours. The CO3 was above 250 milligrams per liter in the inlet and 52 in the outlet. And we operated the NBR for a month and collected a sample for analysis of the pharmaceuticals and also analysis of the microbial community. So these are the results. What you can see is the NBR inoculated with our consortia was able to remove the 5 target pollutants, and in addition was also able to degrade chemically related pharmaceuticals from the wastewater. So in conclusion, we can say that we could manage with this approach to achieve a stability of adapted community over time, that those microorganisms are capable to recover the degrading capacity after stress has been applied. We are community composition and the microbial diversity also affect the degradation efficiency of our systems. And we are convinced that membrane bioreactor operated at infinite sludge retention time can be useful to accelerate the evolution of the microbial community. And this enables us to go and pharmaceuticals even at a microgram per liter concentration, and to demonstrate the principle in real wastewater metrics. So with that, I thank you for your attention.
Yeah. Okay, this was the end of the first session, which was focused on wastewater, and now what is appearing on the screen is the QR code related, from which you can access the questionnaire about this kind of technologies that we developed in NYMPHE. I would like to know if there are questions from the audience or from remote. Yeah, okay, maybe you need to come here.
So I was curious about how the system of the geobacteria that the pellet works. Is the metabolized like acetate or any candidates? Is it going into the pellet or how does it work?
Thank you for your question. What we basically do is we encapsulate, like we bioremediate the microbe. We material, and that material is partially easy to break. So it's compact. We know that there are particles that are bigger and are compact, but the pellet itself, whenever the water passes through it, it partially breaks. So in a way, it's releasing microbes that are actually there along the way. Okay, so it was I didn't have time to explain in detail, but that's how the microbes are, let's say, leaving the pellet and colonizing the rest of the delivery system. Exactly. So that's why we only see it on the surface of the system. In a way, it's a special material that they develop in order to be broken while the water passes through it and then release the microbes that are left inside. Thank you for the question.
Okay, we also have a question from remote, and I do not know to which is the what technology is related, but I guess it is applied to both of you. They want to know if, since the wastewater can have different features, different characteristics, how this can affect the technologies you are proposing and the quality of the water it comes out with.
Thank you for the question. It's an interesting one. Of course, each water is a very different world. That's why we have to do a lot of tests beforehand. But it's true that the nature of the water affects, obviously, in the community that we have to work with. It will be a different community, so we have to pre-seed and pre-inoculate with communities that we have already in our pool of communities. We think that suits better to that water, and it's crucial to know the water beforehand, the kind of water. Human wastewaters are pretty much very similar together or between each other, but the student industrial waters are very, and that's a challenge, and that's something that makes us to really do a lot of tests beforehand. Sometimes it's even not enough. 1 month of testing is even necessary or required 4 months of piloting beforehand to develop a community that is strong enough and effective enough to feed the water.
Philippe, maybe you want to add something about this? The quality of the wastewater, of the initial wastewater, can affect the performance of the bacteria that can remove pharmaceuticals.
I think we get to know more about the bioremediation of the wastewater composition on the degradation of the pollutant. And what we also know is that the type of pollutant that we can find in wastewater may influence the degradation of the pollutant present.
Okay, thank you very much. More questions? Okay, maybe I will add something because I guess that's something that stakeholders and especially end users would like to know about, especially the method technologies. Is how much can you scale it up, and down. So for instance, can you make it bigger enough for a city, or can you make it small enough to be applied to a single house, single home?
Thank you, Elena, for the question. For us, it's better to scale down than scale up. Obviously, CapEx for us increases a lot whenever to address bigger populations. So in a way, we are limited by that. So in urban, I think our limit is 2,000, 3,000. But on the other hand, to scale down for us, obviously depends on the water as well, but it's very easy. And we are capable of, doing small devices for single house solutions, and 1 cubic meter, even half of a cubic meter of the system, you can pretty much treat water for 1 pound. So we are obviously getting better, trying to achieve smaller volumes for more people and more, say, harder water. Yeah, for us, I think our drawback or our limitation is more upper than down.
So it may become a commodity for country houses. Okay.
So thank you very much for this session. But before we go, we would like very much to invite you to join our questionnaire, especially our attendees online. So please use the QR code or the link to the questionnaire. It will just take like 3 minutes of the precious time. And the question and the result at the end in a few minutes, and we'll see just the basic results.
Okay, I just want that at the end of the 3 sessions, we will have a moment in which we will discuss the results of this questionnaire. We will see the aggregate results and see how the people have reacted to the technologies of NYMPHE. So please, also for the people connected from remote, stay connected. We have a 30 minutes break, and then we will be back.
Environmental contamination is one of the most pressing challenges our planet faces today. Unsustainable production and consumption flood nature with plastics, fuels, pharmaceuticals, and other chemicals. Only 37% of Europe's surface waters meet ecological standards. An estimated 2.8 million sites across the European Union may be contaminated. Toxins seep into our air, water, and soil, endangering life and limiting community well-being, but also leads to economic damage. Bioremediation uses living organisms, microorganisms, enzymes, plants, and animals to break down harmful contaminants like oil spills or microplastic waste, which accumulate in the environment into less toxic substances. This approach reduces pollution impacts while conserving ecosystems and boosting biodiversity. Why bioremediation? Bioremediation uses natural processes to clean pollution without harmful chemicals. Microbes break down pollutants into harmless substances like water and carbon dioxide, up to 50% cheaper than conventional cleanup methods. Preserves soil health, reduces erosion, and aids ecosystem recovery. Restores environments while supporting the circular economy. However, bioremediation requires a careful selection of organisms and their assembly that addresses certain types of contaminations. That's what NYMPHE is all about. NYMPHE is developing strategies to restore soil, sediments, and water. Our technologies are wastewater technology, soil technology, microbiomes modeling technology. To demonstrate bioremediation efficiency, NYMPHE will test the biological systems at 4 heavily polluted sites: Erpe River, Germany; Ferrara Province, Italy; Falasana, Western Crete in Greece; Alcalá, Spain; Leopoldov, Slovak Republic. We have one bold goal: to remove 90% of pollutants from contaminated sites. Hopefully, this will only be the start of a less polluted environment for all organisms to thrive in. Bioremediation is not just a method, it's a promise of cleaner, greener future.
So let's start with the second session of this workshop. This will be about the treatment of polluted soils in this case. So I give the stage to my colleague, Professor Borin from the University of Milan.
In the NYMPHE project, I'm Sara Borin from the University of Milan, and in the NYMPHE project, we focused on the exploitation of the assembly between plant and microorganisms for the bioremediation of hydrocarbon contaminated soil. So the assembly between plant and microorganisms is a natural assembly. So plants like us, like humans, are associated with the microbiome. So the plant can be defined as an holobiont that is composed by the host, the plant organism, and by associated microorganisms. Plants mostly recruit their microbiome from the surrounding soil and select a specific bacterial community that can be beneficial. So the plant microbiome has become a reservoir of genes and additional functions that improve the host. Which function? They can protect the plant, coming in competition with the pathogens, they improve plant nutrition, improve plant growth. But in contaminated soil, the functions that are more important and interesting are the biodegradation of the pollutant, so the function in which also we are interested, and the protection of the plant from the harsh environmental conditions. So often contaminated soil are also phytotoxic, so challenging for plants. And plants, when they are challenged, so when they experience a kind of environmental stress, cannot escape like animals from the challenge, but have been described to change a bit their metabolism, changing the array of secondary metabolites with a specific function to interact better with beneficial microorganisms. So this was called a cry for the health of beneficial microorganisms. So this mechanism that is well known and has been described is hypothesized to be also important in bioremediation. So in polluted soil, so under the pollution stress, the hypothesis is that plants change the chemistry of the compounds that are released from the roots, that are called root exudates, and this change can be biostimulant toward the degrading community in the soil. So rhizoremediation should reside on this mechanism. So rhizoremediation is defined as the soil bioremediation in the rhizosphere niche. The rhizosphere is a specific niche with a very high microbial activity and microbial abundance compared with the surrounding soil, and is the soil that is adherent to the plant roots. So rhizoremediation is exploiting the natural assembly between plant and soil microorganisms that may contain pollutant degraders. So the plants in the rhizosphere are biostimulant of the soil microbial community and can enrich degraders that in turn help the plant to cope with the soil phytotoxicity. This is the ecological model. But we can also engineer the assembly between plant and microorganisms by inoculating the plant with selected microorganisms that can help the holobiont to perform better the biodegradation of the pollutant. So which are the important factors that are important in this assembly between plant and microorganisms? So the plant partner should tolerate the contaminant because it must be able to grow in the contaminated soil that can be phytotoxic. Should have a higher root biomass and soil exploration potential in order to maximize the rhizosphere niche in the contaminated soil where the degradation can occur, and should stimulate degrading microbes. So recruit degrading microorganisms from the surrounding soil and enrich them in abundance and activity. The microbial partner, especially the inoculated strains, should tolerate or better degrade the contaminant, so it's better if they are degraders. They should colonize the plant rhizosphere in order to establish this relation with the plant, should improve the growth of the plant, so has plant growth promoting activity, and help the plant to survive better in the soil, so maximize the rhizosphere environment. And an important function is also if the inoculated strains are able to trigger the autochthonous community, so to stimulate even other bacterial degraders to improve in activity and abundance in the rhizosphere. So these were the aims that we explored during these years of the NYMPHE project. We focused on an industrial site, the ER site, that will be also described in the next presentation, that is contaminated by petroleum hydrocarbons and some chlorinated hydrocarbons and metals. We chose the plant, so the plant was a sunflower that was described in the literature and had been applied in other hydrocarbon contaminated sites with good results. And the most mature results are using these two microorganisms, these two strains, Rhodococcus josti B12 and Acinetobacter albaceticus Pri 320. These were bacterial strains that were isolated in another industrial site from the rhizosphere of autochthonous plants, so plants that were able to cope with the presence of the contaminant. These two strains were very good in colonizing sunflower, were degrading, were able to grow on different hydrocarbons and as a unique carbon source. Here you see some data about decay degradation in vitro in 72 hours. So they were degraders, they were plant colonizers, and also they had an activity in plant growth promotion. So when inoculated sunflower was even improved in the growth by the activity of these two inoculants. So the first question that in which we worked is sunflower that was used in parallel sites a good candidate for the rhizo-remediation in this site, in the ER site? So we tested if sunflower is able to grow in this contaminated soil, and we also tested if in the contaminated soil sunflower is releasing, so it's changing the chemistry of the root exudates, is releasing the compound as a response to the soil contamination, in line with the cry for the help mechanism. So the first critical point that we had to face is that ER soil is phytotoxic. So here you can see sunflower that when it's growing in the contaminated soil compared with soil from the same site but not contaminated. So the contaminated soil is affecting the plant growth, but not only on sunflower. We tested also alfalfa that was originally we planned to work on both the plant, but alfalfa couldn't grow at all in this soil. And there are also some results of another group in NYMPHE from the Czech Republic. They are working with tobacco on the same soil, tobacco genetically modified in order to degrade hydrocarbons, and even tobacco is experiencing soil phytotoxicity. So this is the first critical point that may affect every setup of rhizo-remediation. Plants need to grow in the contaminated soil. So we tried some mitigation actions. So one mitigation could be the addition of the soil with an organic amendment. In the graph, you can see that when we added to the ER soil some compost, sunflower was able to improve its growth. So the addition of compost was helping. But also what was helping was the inoculation of the plant with not with bacterial degraders, but with plant growth promoters. You see in the picture that the inoculated plant is more developed than the control plant that didn't receive these beneficial microorganisms. So with these mitigation actions, we could set up the experiments, and so we can think that sunflower is a good candidate for rhizo-remediation in this soil. So the second aspect was, is sunflower activating the cry for help? So is it changing the root exudation when it is growing in the contaminated soil? So we set up a special experimental protocol in order to collect the compounds that were released by the plants after the growth in the contaminated soil and compare them with the same compounds released by sunflower growing in not contaminated soil. The soil was more or less the same, so it was coming from the same site. What was different was the level of contamination. We applied a metabolomic analysis on the two different root exudates, and these are the results. So there is a difference. We could annotate almost 1,500 different metabolites in the root exudates, and we identified 86 of these produced. So 86 were produced in statistically significant higher abundance, only when the plant is growing in the contaminated soil. These metabolites were secondary metabolites, was comprising coumarins, flavonoids, and terpenoids that are known in the literature to be involved in the stress plant response. So the sunflower, when it was growing in the soil, was producing, changing the exudates, and this differential compound, the hypothesis is that they may be biostimulant of degraders. It still needs to be demonstrated, but these compounds are very good candidates, and we are working on this. So the next step is to make the assembly. So to demonstrate if sunflower combined with to identify degrading bacteria can have an effect on the contaminant concentration, but also on the overall microbiome community in the soil, in the rhizosphere of sunflower. So we described the dynamics of the soil microbiome diversity, biostimulated by the plant and bioaugmented by the addition of Acinetobacter or of Rhodococcus. So this is the experimental setup. So we analyze the unplanted soil planted with sunflower without any bacterial inoculum, or soil planted with sunflower inoculated with Rhodococcus or with Acinetobacter. After 7, 14, and 21 days, we collected the rhizosphere or the soil in the unplanted pot and analyzed. The first analysis was the overall diversity of the bacterial community. So here we can see very well the biostimulation activity that is given just by the sunflower. So I have to have this. No. I will describe. So the rhizosphere are different from the unplanted soil. This means that the plant, the sunflower, is able to change the structure of the soil microbial community. But within the rhizosphere, we see that the rhizosphere that were inoculated by our strains are different from the not inoculated rhizosphere. So this is telling that we have biostimulation by the sunflower, but also the bioaugmentation by the strains is able to differentiate the bacterial community. Let's look at the degrading community, that is the function in which we are most interested. So to give an estimate of the degrading potential, we quantified on the soil metagenome some genes that are modern genes known to be involved in pathways for the degradation of alkanes and or aromatic hydrocarbons. So also here we see the biostimulation. So the not inoculated plants or the initial soil have negligible content of these genes, but the presence of sunflower and more the inoculation of the degrading microorganisms significantly increase the concentration of these genes, with some differences, but we demonstrated that we have biostimulation and also bioaugmentation is important. What happened to the hydrocarbons? So it was not easy since the quantities of soil were very small, but we could quantify the light hydrocarbons and heavy hydrocarbons in the rhizosphere at the end of the experiment. And the results are saying that Acinetobacter had an effect in decreasing significantly light hydrocarbons, while the second strain, the Rhodococcus that we inoculated, was effective in reducing significantly the heavy hydrocarbons from the rhizosphere soil, compared with the rhizosphere of plants that were not inoculated. But the question is, are the two strains, the inoculants, the only responsible for this degradation, or their effect in changing the structure of the community triggered for the improving abundance and activity of other autochthonous degraders? The answer to this question is not easy and not simple to be verified. So the first, we try to find an evidence by making a simple differential abundance analysis comparing the relative abundance of the common families that we have in not inoculated sunflower with the families that we found, the abundance of the family of the same family that we find in inoculated sunflower. So the first result that made us very happy was that in inoculated plants, we have several order of magnitudes higher the family of Rhodococcus and the family of Acinetobacter. This means that the inoculant was able to establish very well in the rhizosphere of the plant, and this is a good starting result. But then we see that the two different inoculants had a different effect on the common families between inoculated and not inoculated plants. Rhodococcus, mainly on the common families, led to a reduction of the autochthonous families. So it means that Rhodococcus probably came in competition with some microorganisms that were present in the rhizosphere microbiome. While Acinetobacter was more social, so some families in the Acinetobacter inoculated plants decrease in abundance, so they are in the left part of the graph, but some other families increase in relative abundance compared with not inoculated plants. So the inoculation of Acinetobacter triggered or biostimulated even the increase in relative abundance of other autochthonous families. We still don't know if these families contain degraders, but this is the first evidence that the bioaugmentation not only led to the increase of the inoculum in the rhizosphere, but also is changing the autochthonous community with the further biostimulation effect. And this is very interesting since potentially can further improve the degradation potential in the overall microbiome of this engineered holobiome. So to conclude, we have demonstrated all of the prerequisites that are important in setting up a bioremediation approach. So in particular, we identified plant growth promoting bacteria and organic amendment as promising, so potentially able to decrease the phytotoxicity of the soil. That was the initial critical point. We demonstrated that sunflower is releasing differential compounds in the soil when it is growing in the contaminated soil, and that could be potential biostimulants of the degrading community that can be used as soil amendments without the microorganism. And we demonstrated that the assembly between sunflower and these two strains reshaped the overall bacterial composition in the rhizosphere, increased the degrading potential in the rhizosphere, and led to a partial removal of hydrocarbons, which are the next step. So we will increase the size of the experiment. So from a few grams of soil, we will work in kilos of soil, seeing if the scale up will work with the same efficiency. And we will apply the organic amendment, and we will also try to plant, if the plant cycle is shorter, we will plant a successive plant in order to improve the effect with subsequent planting. In the meantime, we are testing the metabolites that we have identified as produced in the contaminated soil produced by sunflower. So we commercially available, we got them and we are testing them on some bacterial degrader, trying to find a compound that can stimulate the degrading microorganisms, so can be used as a soil amendment. So with this, I thank my group from the VIM lab and the groups that collaborated in this work. And
so this will be a shared presentation between the University of Bologna and ENI Rewind, my colleague Maria Alejandra Decima. We have been working we are working in the NYMPHE project on assemblies of microbial assemblies for targeting the remediation of subsurface soils contaminated by mixed hydrocarbon pollutants. Okay, this is so just to show you that our target site is an industrial soil where the soil below 1 meter depth and the groundwater as well are contaminated by a mixture of different hydrocarbons, monoaromatic hydrocarbons, so some polycyclic, very little concentrations of polycyclic aromatic hydrocarbons, and then of course also aliphatic, light and heavy hydrocarbons, plus in some locations also some chlorinated ones. So given the, let's say, the current situation in terms of pollution complexity, the approach we are following in NYMPHE is to actually bioaugment the contaminated matrices through an assembly of microbial communities that each are targeting specifically one class of this pollutant mixture. And in particular, we want to perform so-called autochthonous bioaugmentation, introduce at higher concentrations those microbes, those that have been isolated from the contaminated site, since the basic might increase the viability, survival, and use contaminated site, and so promote better bioremediation. So actually, the activities, and we are going to conclude in the next months. First are related to the attainment of microbial control or pure bacterial strains that are able to degrade polluted site. This, of course, at the laboratory level, where we also and assess how assemblies of these microbial communities that can target the pollutant mixture. First, in very simple, let's say, far from reality conditions, so in mineral medium, but then, of course, going to the real soil. And then finally, in the presentation, the colleagues from ENI Rewind will show you the test, how we are performing the test in the field. Okay, this is my problem with Mac, I'm sorry, which is every time or very often, but it is visible. So this is just to show you very briefly that during the very first part of the project, we have enriched different microbial communities that were able to target or to degrade some polycyclic aromatic hydrocarbons, in particular naphthalene, phenanthrene, and let's extend anthracene. Other microbial cultures that were able to degrade, let's say, the total petroleum hydrocarbons. We were in this case considering diesel fuel, and not only acting on the light, but also on the medium, and partially also on the high molecular weight fraction of the mixture. And different cultures degrading each of the BTEX mixture with, however, a lower degradation rate for the xylenes. Anyway, even these cultures tested, I was mentioning in a preliminary way, so a very simple situation, mineral medium with a pollutant, combinations of these cultures to make sure that they can be active together and that they are not suffering inhibitory effect from the presence of other pollutants. So the 4 cultures degrading the single BTEX compounds were also shown when put together to degrade simultaneously the four BTEX, and when they were mixed and co-incubated with the polycyclic aromatic hydrocarbon degraders, of course, in the presence of all pollutants again, we could confirm that all the target pollutants in the assembly were degraded with a very similar efficiency and rate. Actually, this was observed also when assembling the 4 BTEX degrader with the total petroleum hydrocarbon degraders. So, for example, in the graph below, you can see that the degradation of aliphatic hydrocarbons is the same when we use the only dedicated specific culture and the 2 cultures together. Regarding the BTEX degradation, actually BTEX degradation was even faster when we were mixing the BTEX degraders with the total petroleum hydrocarbon degraders. So these cultures can be assembled in principle without any potential inhibitory, cross-inhibitory effect. This was not observed, sorry again for the black thing, this was not the case when we were combining the total petroleum hydrocarbon degraders with polycyclic aromatic hydrocarbons. So what we observed is that the degradation of those aliphatic hydrocarbons, or the degraders of the aliphatic hydrocarbons, were inhibited by the presence of naphthalene, phenanthrene, and anthracene, which is telling us that these cannot work together. However, let's say by inoculating the system with the total petroleum hydrocarbon degraders after the degradation of polycyclic aromatic hydrocarbons has taken place, then allows also to target the degradation of the aliphatics, which means that their degradation is not leaving behind any product that is further inhibiting the degradation of the degraders of total petroleum hydrocarbons. So after this, let's say, preliminary test, we have moved to, let's say, performing the assembly in the real soil collected from the site. In this setup, we were, of course, adding some nitrogen and phosphorus sources to correct the carbon nitrogen phosphorus balance in the soil, incubating that could also allow us to perform respirometric measurements. And we were having controls that were not inoculated, and our tests that were inoculated with the selected cultures. Here I show you only the inoculation with the degraders of polycyclic aromatic carbons and BTEX, because the inoculation for the aliphatic degraders must be performed only when pHs have been almost fully degraded. So the graph below shows with the orange line that the microbial activity in the inoculated system, so in the biologic assembly, is higher than the one we observe with the native microbiome only. So we expect higher microbial activities and degradation rates, which was actually what we observed both on the BTEX and on some polycyclic aromatic hydrocarbons. In particular, on the top right, you see that xylene, on the top, you see that all the BTEX are degraded by the indigenous community if you provide oxygen and nutrients, but that with the in the inoculated system, both benzene, toluene, and ethylbanes are degraded much faster. This lower concentration at time zero, actually, we believe it is fast biodegradation because this sampling was performed 24 hours after inoculation. So the xylene degradation, xylenes are degraded very slowly after 24 hours. Still, their initial concentration is the one, the original one. After 24 hours, the other beetles have been already quite fast depleted. Similarly, this is naphthalene, phenolphthalein, and anthracene. This is a picture taken at day 59, so 2 months after the inoculation. Let's say the system is working fast on naphthalene, much more slowly on the other two. So we need to prolong, let's say, the incubations. However, let's say, given these promising results, those cultures have been already selected and produced, cultivated in larger volumes using a 15-liter bioreactor. So this inocula have been produced to be applied directly at the site. And now, let's say, I leave the floor to my colleague that who will explain the question of the site.
Good morning, everyone. Okay, now we see. Okay, here we can see in this slide some steps. Phase 0 and phase 1. Phase 0 was about the site characterization because, as the professor showed, in the beginning we made a characterization. So that was already drawn. The phase 1 was about the microbial inoculation preparation that was already explained by the professor. And now we are going to see what happened at in the file activities. So we are going to see phase 2 and phase 3. What's the difference? In phase 2, we only test the enhanced remediation aerobic. I mean, we only inject in that part some a compound that release oxygen, And in the phase 3, we are going to test the same, but we are also going to make a bioaugmentation. So we are going to compare what happened if bioaugmentation makes an enhance the biodegradation or not. So let's see. Okay, this is the phase number 2. Here we can see the part of the file where we test the activities. And here we can see that the target contaminant in this part were hydrocarbons. The selected technology was aerobic enhanced bioremediation. So we are going to inject a commercial product that releases oxygen in a slowly way. And then we have selected and we have tried a new piezometer that it is called NYMPHE. So here we can see what was done in the file. We have tried the piezometer that is the point here, and surrounded we inject this product. So the points, the yellow cone, where we injected those 3 meters away. So let's see the other part. Okay. There are some photographs of the file activities. In this photograph, we can see where the moment where the line 1 was reading. The second part, we can see the soil that was extracted from the drilling that was useful to characterize the contamination. And the third photograph, we can see here the groundwater samples. Here, okay. In this slide, you you can see the moment in which the ORC compound was injected. In the first photograph, we can see that the product first was mixed with water. Here we can see. Then was injected, and the technique that was used was by the push. So the machine, your cross machine, will inject this product from 4 meters and 2 meters from the ground level. Okay, and these are the results. The results until now, because the experimentation hasn't finished yet. So, the first part, we can see that in the first graph, the upper graph, we can see that the solar oxygen and the potential redox, how they change in the time. So, the vertical line divides the baseline and the monitoring after injection. So, we can see this oxygen, we can see that after the injection, the oxygen grows and then slowly decreases. The potential redox pattern is more or less the same, also increase after the injection and this is something that we expected and we wanted, because in that way microorganisms can grow. So then we monitor also the concentration of the contaminants. So here we can see that the total hydrocarbon emission of the trend, the decrease over the time. You can see here there is a line that shows that slowly is decreasing. Also, you can see that the most contaminant here, the maximum concentration was about the hydrocarbon that was the shorter chain. So here, this slide is so similar to the other slide, because the scheme was so similar, but here we also inoculated the microorganism. So here we have line 1 is the piezometer where we checked the ORC compound, only ORC compound. That was a result that we have just seen. And here in line 3, the other piezometer that it is near line 1, so the area is the same. But here we inoculate, we injected. Also in this scheme, the same quantity of the ORC compound, and also we injected the inoculant that was prepared by Unigo. So this was the inoculant was just inoculated more or less 1 month ago, so we don't have results yet. So now we have to wait the next monitoring campaign to evaluate the concentrations. So next steps. In phase 2, that was only the ORC injection, we have to continue with the groundwater monitoring campaigns. And in 3.3, we have to start the groundwater monitoring campaigns, and then we have to compare both results. So, as a conclusion, we can say that the PPX and IPI are the components that have been shown in degradation during the life when the consortium with bacterial consortium was prepared and tested during the lab. That was the Professor Sanadroni had shown. And regarding the file activities, we have seen that following the injection of ORC, we have seen a decrease of the concentration of total hydrocarbons in line 1. Nevertheless, we have to continue monitoring to have an overview of the technology. And regarding phase 3, we don't have results yet to show, so we have to wait. Okay, and this is the group that any rewind and we will so thanks. So thanks to the speaker for this interesting presentations. Now you can see another QR code, which is different from the previous one, and this is will connect you to a different questionnaire, which is more focused on soil bioremediation technologies and your perception about it. So please, both here in presence and from remote, fill in the questionnaire. In the meantime, there are questions from the audience. Okay, so maybe I'll break the ice. I wanted to ask maybe something which is interesting for stakeholders and then users. Sarah, if you plant this sunflower on a soil which is polluted like this, can you use the oil or will the it be contaminated?
It's a paramount problem every time that we try to apply remediation. There are efforts of all the compounds, and especially for highly hydrophobic compounds with high molecular weight, scarce and not common. But anyway, we should first check it, and I would say.
Okay, thank you. So we will need to use the sunflower as a decoration. Okay. Someone else? Yes. Can you please come here because.
Hello, I have a question for the vaccination. How it was to scale up the microplastics?
Yes, it's quite, we say, complicated when you are, of course, let's say this way. We were assessing different microbial cultures, and so the scale up was done growing each culture individually. Okay. The problem is that in some cases we are dealing not with pure cultures, but with microbial communities, and we know that even if they are might be quite stable, they are changing. They may change composition. So what we have done was first, let's say, to try to keep as much as possible selected conditions during the fermentation. In some cases, of course, we couldn't use the pollutant as substrate. So we have selected an alternative substrate that may be, for example, an intermediate of the degradation pathway, in order to keep, let's say, not the full, but at least a partial selection on the degraders. Of course, when doing this, showing the results, we have observed, we have characterized the community, we have observed a change in the composition of the community. But then, when this community is brought back under selected conditions on the substrate, again, we observe it's a change in the composition that was the community was going back very close to the original. Actually, this is what we have done in this, let's say, cultivation procedure. After growing, let's say we were testing the degradation, and the recovery of the, let's say, original composition.
More questions? Maybe I have a question for you that might be of interest for someone at home. So, what is the foreseen timeline of an experiment like this? What would you suppose to be the timeline for completely remediate?
You mean
Using microbes
at the laboratory or the
in the field?
in the field?
Can we make some provision?
To me, it's quite difficult to say. Of course, we are expecting, or we may expect in the field, lower, let's say, kinetics, lower rates. We are not in a controlled environment. We can, of course, to adjust and control some parameters like oxygen availability, nutrients availability, but of course we have much higher variability of many environmental conditions that we cannot control in the field. That will of course increase the time needed to achieve the same, hopefully the same degradation results that we can observe in the laboratory. So, for example, I'm expecting that if naphthalene and other polycyclic aromatic hydrocarbons are degraded in a couple of months in the lab, this would take, let's say, much longer. Hopefully not very much, but probably few months more to be achieved in the field. But I think this is normal when you work under conditions that or in systems that can control very well many environmental conditions.
Question from the audience?
We will let you know by the end of the project, of course, when the pilot test will be completed. And so at the next NYMPHE workshop, we will have the final results and answer to some of these questions.
Okay, so if there are no more questions about the soil bioremediation, I would say that we can move to session 3. And it is a session in which we will explore the effort made by the NYMPHE project to improve the ability of microbes to bioremediate. So first of all, we will explore the possibility to act on single microbes, but then we will also move to communities of microbes and to see how we can help them develop. So I leave the stage to my colleague from the C6 in Spain, Thomas.
Good morning, everyone. Do you hear me back there? Yeah? Okay. So my name is Thomas Aparicio. I work in the laboratory of Victor de Lorenzo in the National Center of Biotechnology in Spain, in Madrid. And today I want to show you our efforts to apply synthetic biology to improve microbial activities in the frame of NYMPHE. So the basic idea of NYMPHE is pretty simple. You can see there. Bacteria are isolated from polluted sites, characterized, and then used back in the polluted matrices to detoxify and degrade the pollutants. But how this happens? I mean, most of the pollutants are new to nature molecules, and so bacteria need to learn how to detoxify. So in the beginning, you can imagine that we have a happy bacteria in a clean environment, and suddenly, the human activity spill toxic in that environment. So, of course, bacteria are facing a dramatic situation, either die or obviously adapt to this toxic environment. So what happened? Of course, you know what happened. Evolution. So bacteria displays a number of strategies to evolve, many, but mainly horizontal gene transfer, so they can acquire new genes, plasmids, transposons, piece of genome from the other bacteria, and of course, mutations that help to shape these new activities and introduce in the metabolic network of the bacteria. So the question, well, at the end, most of the bacteria probably die, but some of them learn, the best students, they learn how to degrade the pollutants. So now they are happy again in a clean environment again. This is pure evolution, right? So the question we want to answer is whether we can mimic and accelerate this process of natural evolution in the lab, so make it faster for our purposes, of course. So, because remember that the bacteria that NYMPHE is isolating already have activities. The idea is, can we increase the mutational rate and select in a given pollutant those students that became really, really experts on degradation? Well, this is the question that we're going to answer, and we propose a hit and rank strategy that I will explain now. So the idea is to generate a plasmid device. Plasmid is a small molecule that can replicate in bacteria, and we can modify easily in the lab, right? So that is transmitting this plasmid into a bacterial community that works great, trigger accelerated evolution, and then remove the plasmid. So we have a community or strain that is performing better. The idea is pretty simple. It's just mimicking the natural evolution faster. So for that, we need a number of genetic traits that basically we need a plasmid that can replicate in bacteria, of course, and can be self-transferred from bacteria to bacteria. We need genes that foster mutation, of course. This is the basic thing. An expression system to control the activity of those genes, and to close the circle, a self-cure mechanism to remove the plasmid device and, let's say, erase any trace of genetic manipulation. So let me explain the functional blocks one by one. We already have this hemating, it's a plasmid we developed years ago, and it's able to replicate in most gram negatives. And it is able also to be transmitted from bacteria to bacteria because it contains a conjugative function. So when a primary donor is introduced in the community, the plasmid jumps upon cell-to-cell contact, jumps to another bacteria. And that one, the receptor, becomes conjugative. So when it's close to another bacteria, the plasmid can jump. So this plasmid, let's say, can spread in the community by itself without further manipulation and conquer the whole community. So it's a good way to transfer activities from community. The very keystone of this technology are the mutators. We want to increase the mutational state of bacteria. So we selected the so-called basic. So they are genes, adenosine cytidine deaminase and adenosine deaminase. The first from rat, the rodent here, correct? And the second from E. coli. So these genes encodes enzymatic activities that are very simple. Basically, they deaminate nucleotide. So they transform C on things and A on this. Basically, they work very nice in many, many organisms, not only bacteria, also mammals and plants and everything. So they're very orthogonal. And we fuse both activities. So the idea is expressing this is in bacteria. We can stimulate many changes and a high variability and diversification in bacteria. of course, we want to control this activity. So the idea is using a temperature-inducible system that we already developed years ago. Basically, at low temperature, the system is off, so it's not expressing the antigens. When we rise up the temperature even for a short while, the system is on, and then the genes are expressed and the mutations happen. So it's very easy to use. 30 degrees, 37 degrees, activate or shut down, or activate the mutation. And the very last thing, but also important, is to remove the genetic device that we want to use. So in general, plasmid use a replication protein to initiate replication, so the plasmid is replicating in the cell and stay there in the population. And this protein in general is expressed all the time, so the plasmid is replicating all the time. So we want to use a so-called tolerance switch. It's a quite complex genetic circuit. I'm not going to explain, but basically, if we put the red protein under this tolerance switch, we can play with two inducers, which are simple chemicals and cheap chemicals. So if we use inducer 2, the red protein is expressed, the plasmid replicates in the population. If we remove that and put inducer 1, what happens is the red protein is not expressed any longer, the plasmid doesn't replicate, so when the bacteria duplicate, the plasmid is lost after a few genetics. So this is the way to remove the genetic device. I'm not explaining many results, it's just a glimpse. In the upper part, you can see the application of this technology on single strains, E. coli, Salmonella Futia, Aeruginosa, and some soil bacteria. So what we measure is the mutational rate. I don't know. No. Okay. So when there is no plasmid on the bacteria, the mutational rate typically is very low. It's 10 to -8. So few mutations are happening in the bacteria, but when we apply the system, you can see that the mutational rate increased dramatically. It's increasing for like thousands of times. So all of them became really young. And in the lower part, you can see the sequencing of the genome, some of those bacteria. So the blue line is the genome, and the red dots are the changes we found in the genome. So you can see that different isolates show a different profile of many mutations along the genome. So we can say now that we are producing or fostering variability in bacterial populations. So now the hard part is ongoing, and currently we are trying to evolve a number of NYMPHE isolates to improve the degradation of hydrocarbons, plasticizers, common drugs like enalapril, diclofenac, and pesticides. And I hope that in the close future, we'll have some good students that are doing better and can use them in the depolluted matrices. So thank you so much for your presence and attention.
And for our last presentation in this session, microbial modeling and management to improve bioremediation performance by Silvia Petros from Bloomington, Netherlands. And we have Sylvia online. Okay.
Hi, good afternoon, everyone. Yeah, thank you for the opportunity to share our work. I'm Sylvia Catreu from Blossom Microbial Technology. And today I will be sharing how modeling of microbes and management offer new opportunities to enhance bioremediation performance, drawing basically on our work in the NYMPHE project. I will show what lies behind the modeling soil microbes and what the goal of driving cleanup and accelerating ecosystem recovery. When we talk about the microbiome strategies applied for soil cleaning and regeneration, we start with the soil itself. Every single gram of soil contains millions of microbes. We have already heard a couple of previous presentations about this. Microbes like bacteria and other types And this is an incredible density of microscopic life that means that soils are not passive, they are dynamic ecosystems. Many of these microbes carry specialized enzymes that can degrade pollutants from hydrocarbons to pesticides. Others interact closely with plants, for example, by modulating heavy metal uptake. We have heard about this in a previous talk, which can either reduce toxicity or help plants tolerate contaminated conditions. So if we think about it, soils have already an intrinsic capacity for self-remediation. And they are like natural cleanup systems that is constantly working in the background. But in most polluted environments, this natural capacity is either too slow, too weak, or disrupted by stress conditions. I think it's good to remember that our big challenge is to unlock and to optimize these microbial functions. Was already presented by Thomas before that this can be quite tricky when it comes to bacteria that is present in these polluted environments. And that means that not only understanding who these microbes are, but also how we can support them, feed them or guide them so that they can perform more effectively in cleaning up contaminated environments. So in other words, I would say it's a functional microbiome is essential for a self-cleaning ecosystem. And yeah, the question is, how do we make sure that self-cleaning potential is fully realized in the sites we are trying to recover? So as I said, soils already carry the power of self-remediation. And inside any soil microbiome, we can find microbes with different roles. Some of them are cleaners, microbes that can degrade pollutants such as dioxins, pesticides. Others, they have unknown functions that we are only beginning to understand. And some can also be deleterious, meaning that they block degradation or compete with beneficial microbes. In general, we can think at this like a control panel of functions. Certain microbes are switched on, while others are switched off, depending on the conditions in the soil. So if the wrong switches dominate, remediation is slow and ineffective. As we are busy with this, we try to rebalance this control panel. And one of the ways to do this is through prebiotic cocktails. So instead of adding foreign microbes, we selectively feed the right ones that are already present in the soil, the ones that carry the useful functions for breaking down pollutants. So by doing so, we can switch on the degradation of key contaminants. So, and while switching off competing or blocking activities. The result is an what we call optimized microbiome that works more efficiently in the self-cleaning ecosystem. So, but we still have the question that how do we decide which functions to activate and which interventions will work best at a given site? And this is where modeling and digital twin approaches come in. The starting point is a relationship between the microbiome and the bioremediation performance of this microbiome. So by studying which microbes are present and which pathway they use, we can build models that connect community composition with degradation outcomes. And once we have that model, we can run in silico experiments. So instead of testing 1 or 2 treatments in the lab, we can simulate millions of possible interventions, different combinations of pesticides, additives, or environmental conditions. In this way, we build a model that helps us to narrow down which options are most promising. And from there, we move to designing activators. These are basically tailored interventions that selectively stimulate selectively stimulate the microbial functions. So, for example, boosting hydrocarbon degradation or accelerating pesticides breakdown. And from here on, we move to the predictions that are validated in real systems, both in the lab and in the pilot sites. And this step is still crucial. We have heard also today that it tells us not only whether the model is accurate, but also how it needs to be fine-tuned for the different pollutants and soil types. So the digital twin strategies can give us the possibility to model, predict, design, and to optimize the validation and to guide further the soil recovery. And I'm going to give you a few examples of what we actually do in NYMPHE. So here is an example of how we apply the digital twin approach in practice, focusing on soils impacted by pesticides and microplastics. The challenge in this environment, as we heard already from Giulio, is that degradation requires several processes working together, like redox reactions, fatty acids metabolism, or aromatic degradation pathways. And these pathways are carried out by specific microbes that are often already present in soil, such as Sphingomonas, Bulgarderia, Chromobacter. And what we do in NYMPHE is that we use the modeling to identify the candidate, what we call candidate microbiome enhancers. Essentially, these are compounds, prebiotic compounds that could stimulate the right microbial activities. So we have here on the left examples like acetate, amino acids, or cofactors that can boost pollutant degrading functions. And on the right is just a bar chart with the ranks of the top 20 biostimulants that were identified for this specific soil contaminated with microplastic. The length of each bar reflects the performance score, which integrates how strongly a compound or a combination enhances the relevant metabolic pathway in the model, and how consistently it supports the microbial growth in the digital environment, and we estimate from that the pollutant degradation. So in other words, the higher the score, the more reliable and effective the biostimulant is expected to be. Another example is when we tested these modeling strategies for hydrocarbon impacted soil. We have heard today that hydrocarbons are among the most challenging pollutants. But applying our modeling approach, we identified a couple of ways to stimulate the right microbial pathways. So we estimate that the results can show a 15 to 30% increase in metabolic flux through key hydrocarbon degradation routes, such as alkaline monoxygenases or beta oxidation. Importantly, this was also estimated by higher biomass accumulation in the digital environment, which indicates a possible stronger microbial fitness and resilience. So the biodegradation expected to increase, as it's visible here in this slide. We got a list of candidate microbiome enhancers, so compounds that can selectively activate hydrocarbon degrading microbes. And some of these are familiar molecules like acetate, succinate, nicotinate, which act as cofactors or osmoprotectants. So the expected increases in degradation flux for each enhancer, for example, nicotinate gives an about a 30% increase in the degradation, glutamate about 28, glycine about 27. So these are substantial improvements that can make the difference between slow, incomplete, and a robust remediation process. Not to forget that equally important is also the shift in microbiome structure, and we have also seen with the experiments, plant experiments presented earlier, that with the right enhancer, we can see possible dominance of hydrocarbon degraders like Pseudomonas and Rhodococcus. While with other enhancer, we can get a broader increase in community diversity and activity. So both outcomes can be valuable, depending also on the site and on the pollutant mixture. So just as like with the pesticides and microplastics, we can use modeling to predict which additives will deliver the most effective microbial response. And this gives us, we think, site-specific and a very good strategy for tackling the contaminated soil. But it comes to the end to the question, why choose bioremediation and why to model it? We think that it's definitely in favor of choosing bioremediation because it works with nature. And we have hundreds of examples of this happening really in nature. And we choose modeling because it allows us to guide and amplify this natural power of microbial communities. So first is the fact that one site is never the same as another. So every soil, if we talk about soils, has its own microbial community, its own chemistry, and its own history of contamination. And that's why one-size-fits-all approach to bioremediation really works. So community context always shapes the response. And here comes the modeling to help with this, enhancing this response. Second, we now have the tools to design precision additives, whether they are natural molecules or let's say like amino acids or organic acids or cofactors, or other type of molecules that can, these additives can selectively boost the microbial pathways as we want. And with this approach, we see increases in up to 20 to 35% of metabolic flux, which directly, we know from lab experiments and greenhouse experiments, that directly translates into stronger pollutant degradation and more resilient microbial consortia. And in the third, sometimes even a small uplift has a big effect. So for example, just a 25% increase in microbial activity can shorten remediation timeframes and reduce pollutant persistence. This is why predictive modeling is so powerful, we think. It helps us to identify the small tweaks, let's say, that creates impacts. So to conclude, I would like to say that bioremediation gives us a sustainable path, and definitely modeling makes it precise, predictable, and scalable. So together we can transform and we can act faster to recover polluted environments. And with this, I will like to thank you very much for your attention, and I hope the connection was good, and I'm ready to take any question in the Q&A session that follows the presentation. Thank you very much.
The session, I guess, for people who are not really in the field, but it is really a vision of the future. There are questions from the audience or from...
Couple of questions, one for each speaker. The first one is about the tool. So my question is whether there is any chance that the plasmid can integrate with the chromosomes. If it is the case, then your system for eliminating it is still working.
That's a difficult question. Yes, this might happen. Well, I mean, we know that it doesn't happen frequently, but this might happen. We have to live with that. If we both a single strain, there is ways to remove from the genome. We have the tools for that. If we talk about communities, it's more complicated. This is something we have to test. We do experiments with communities. That, yeah, with communities, everything would be more difficult, and this might happen. Anyway, I didn't mention that we want to include in the plasmid activities that ensures that if the plasmid or integration is present, the bacteria dies. We can use, for instance, the SAC gene. It is a gene that when it's expressed in normal condition, it's not toxic, nothing happens. But if you put sucrose, the activity of this gene transforms sucrose in levulsurans, which are super toxic. So we are planning to add these additional layers of control to do the best to eliminate the plasmid or the bacteria do something like that. So this is in the Thank you for the question.
I know that you have another question just before, since I forgot. This is another QR code for another questionnaire, which is related to these new tools for microbiome and bioremediation technologies, and I invite everyone to answer the questions. So.
Yeah, the question is for the modeling to the speaker, right? So my question, if you hear me, is whether your metabolic model are only based on genomic annotation, or do you need also some aspects analysis or phenotypic experiment? Because I think that this is crucial for scaling up and to real more complicated PRPs studies.
Indeed, yeah, indeed, yeah. So the origin for any unknown, can you hear me? Yeah. So for the unknown environment, we start with the analysis of the genome. But as the process of, let's say, building the digital twin, detailed analysis of the community there and the most potential community members are included. So therefore, the model is increased. So it's helped by the metabolic informations and all the physiological data that can be collected from the community. And this is the case not only for bacteria. Okay, we know from the general knowledge built around this digital twin that for bacteria it's a bit more easy. But we also include the information from the other microorganisms like fungi or like algae or like small protozoans that they are in this environment. Because the final goal is to have a digital twin that reflects as much as possible the real environment. This is the only way to achieve some results in the end. Yeah.
Thank you, Silvia. More questions? Okay, maybe I have one for Silvia because today, what you did up to now in NYMPHE required a lot of expertise in informatics and in biology and microbiology and biotechnology and so on. But can you see a future in which your model can become more user-friendly, so other people that have not your expertise can use it, such as a software or something like that, putting input, giving some input to the model and obtaining the output, the indication of what they need to do in the field without having your expertise and building the model from scratch.
Yeah, absolutely. This is actually the moment, and it's good that we are here all, because this is the moment in history of bioremediation, let's say, when we move the abstract and complicated process that we do in the computers and in the lab, we move it and we transfer it to the to a real technology that can be used. So all these digital twins are, as we speak, they are already packed and can be packed into software packages, into large language models that are questioned by everyone with normal conversations. So everyone can have a normal conversation using simple conversation words and interrogate these models. And definitely with the advent of this large programming and evolution of the programming languages in this direction, these digital twins become more accessible to everyone. We have passed the time when the genome we talk about genomes of organisms, of plants, of humans. And this genome is not anymore locked and accessible only to the scientists. This genome or any complicated information can be really translated into tools and approaches that everyone can access it. So definitely it's possible, yeah. And it's as we speak, it's happening. Yeah.
Thank you. Something more? No questions from the board? Okay, so I think that Julia, you want to say something more?
Yeah, so I just want to add that probably after we will close this workshop, and as we know, the workshop was advertised and is part of the RemTech 2025 program. And so when we will be at the end in half an hour, and we go and ask one of the available chats, we will already can be able probably to fish out information that was discussed here, and open access is offered in NYMPHE for all these discussions, and everyone can have access to this information. And this is a great achievement for the field of bioremediation, I think.
Yeah, thank you very much. I think that maybe we have to thank all the speakers of today, and we can start a final discussion if someone wants to join me, and we. Thank you.
Thank you very much. So, I'm Andrea Franzetti from University of Bicocca. I am honored to co-chair this roundtable discussion about primarily mostly the result of the feedback that we obtained from the questionnaire. So the panel, so the participating to the roundtable will be the speakers, and the speaker will be also joined by Pavlos Biologon, that is representing RemTech Europe here. He's also a researcher at FISIC in Spain, and also member of the European.
I'm not in Spain actually. I'm in Greece. And that's.
I see. Okay.
Doesn't matter. Maybe I don't licensing. No problem.
Okay. And member of the European Federation of Geology. So as soon as we can see the results of the feedback of the questionnaire, maybe we can do like
Still
working on it, of course. So maybe we can do like this. We will show you the very quickly elaborated results of two main questions, mostly regarding the strength and the weakness and challenges still in the three different fields that we addressed today. So dividing the discussion in the three sessions. So basically, what is done has been done properly. The good news, and also what we have still to do to improve and to enlarge the possibility to apply bioremediation at the real scale. And we will invite first the speaker
to comment on that results, particularly focusing on what they can do in the future, and so to try to cope with the challenges that the stakeholder will highlight in their response. But of course, the discussion is open to anybody can want to participate from here, from the room, and also from remote. I guess that also then all the questionnaires that are quite long and complicated will be further elaborated by and Inina that can give a very more insight into the results. So if I can see, so how relevant do you find the advanced micropollutant removal from wastewater from application? So the answer seems to reveal that the audience is very interested and thinks that it's very relevant in the sector of the people that reply. So I think that we have the right audience to move on to the second question. So we are talking about the methane technology for removing micropollutants from wastewater. And the question was, what do you see as the main technical strength? So the distribution are quite even. So it seems that the efficient removal of natural pollutant, low energy. So these are the options that were present in the question. Scalable and modular design for different plant sites, low maintenance, sustainable and cost-effective process, and easy integration and retrofitting. There seems to be quite all the aspect that has been addressed seems to be relevant and important for the success of the technology. We just want to comment on this, and we go on the challenges. So, if you want to say something on that, please.
No, just comment the fact that it was very homogeneous means that they consider the very strengths of the technology, so it's something that makes us very happy. Just to acknowledge the participation, that's what the thank you to wants to vote it, and let's keep commenting to the.
Yeah, let's move on the challenges, right?
No, this is the main.
This is the strength of the other technology of microbial assembly for pharmaceutical biodegradation. So, Philippe, if you want to comment on that, it seems that in that case the distribution is not so even because the 50% of the answer was regarding the consortium can be adapted to the greater pollutant relevant for the wastewater interest. Instead, the technical strength that was less considerate was the consortium can be trained to deal with concentration, low concentration of pollutants. And the other two questions were more or less the same abundance. So the consortium can be trained to have resistance and biodegradation function, and bioremediation consortium is more stable than with a single strength. Do you want to comment on that? Sorry. So this was the answer of the stakeholder about this question. What do you see the main technical strengths of the microbial assembly for pharmaceutical biodegradation? And so these are the distribution of the answer about the strength. So the main one was the consortium can be adapted to the great target pollutant relevant for wastewaters of interest. So it seems that the strength has been that the stakeholder consider more important is the fact that your consortium can adapt to different target contaminants. Do you want to comment something on that? Okay. So maybe we can move to the other one. Okay, now we are moving to the bad side, to the dark sides of the technology. So the main barrier to an advanced micropollutant removal technology from wastewater. I'm getting older. High cost of infrastructure is the one that has been the question that has been done most. High energy value, uncertainty about long-term effectiveness. So these are the three, let's say the two most answered are high cost
and uncertainty about long-term activities. So I will ask the speaker to address and comment on that.
Yeah, so we mentioned some of these points in the presentation. So during the discussion afterwards, the high cost obviously is something that everyone is interested. That's the economical part. It's true that the costs are high. It depends on how you see it. What is the necessity, the requirement, if the administration is pushing you, is pressing you to solve the situation. So cost is always relevant. So if you really need to implement some solution at the end, you need to see or look it in a long term. In our solution, it's true that the capex could be something at the beginning it's somehow high, considered high. I don't want to say high. Really concerned about. But the strength of our technology is the OpEx, the low OpEx. So in a long term, again, when you do the numbers, you realize that you don't need to spend a maximum maintenance on operating the system. In a long term, when you say long term, it's 5 years or roughly 5 years, you realize that at the end, it's not a high cost. So it's something that I understand, but the audience pointed out this as what option is relative. That's really mentioned. Thank you.
Thank you. Of course, university can have an important role in educating, growing the expertise, adopting these technologies. I think also that, of course, the degree in which one need a specific technical expertise may also depend. For example, with the method technology in terms of operating, and you don't need, let's say, a strong specific technical knowledge, probably more than you would need to operate another current conventional technology. And I think this is also probably a strength in the Maitland technology. Of course, let's say, approaches such as the NDR still are, let's say, innovative approaches, but are based, or should be based on, let's say, technical knowledge that may not be very much different from the technical knowledge you need to, let's say, run a currently used technology, because of course you have the membrane module in addition to maybe conventional wastewater treatment, but still the principle is similar. It's the principle, the detail, the infinite large conventional line that is making a lot of difference in terms of the performances. But I don't think it's adding a lot in terms of, or maybe this was the perception, okay, because this 30–40% means that maybe these technologies were perceived as technically, let's say, complicated to be. So maybe what we should do next is, let's say, try to explain that their implementation and application in the real situations, also large scale, may not be so requiring so much additional expertise. So it is interesting to see this number, because I think it is important indication for us to, let's say, adjust also our approach in describing the technologies.
I also think that this kind of skills should be trained at after graduation. So I think that in that case, also the collaboration between education entities and operators is very important to train people to be very specialized.
Okay, so regarding soil, we have the answer to the question how important is the need to develop soil bioremediation technology. And we have, of course, a distribution which is highly moved toward the important and very important. So this is good because it means that yeah, we have the good audience and the possibility to clean up polluted soil is perceived as important. So regarding what do you see as the main technical strength in plant microbes association, so fighting remediation, rising remediation of contaminated soil. I think that the highest percentages of answer regarded the economic feasibility. So contrary to the technologies related to the micropollutant removal from wastewater, here we have the audience that has perceived the possibility to use plants and microbes as less costly, cheaper. Is it true, Sarah?
Yes, in this case and in other cases that we studied, we choose plants for this reason. So plants can be used in very extended areas where other type of technology may be hampered by the cost of the technology. So using plants, maybe the result is not simply of complete removal, but they can be applied in very extended areas. So the balance between sometimes the lack, or not the lack of result, but it's not exceptional result, but the applicability in extended areas make this approach very promising.
The local value is easily accepted by the public opinion, so probably the public opinion is not perceived as relevant in this case. But the 23% is needed, so as a strength, the possibility to apply the technologies to extended site. Yes.
So just a comment, because I was quite surprised that this first answer, nature-based solution, not really difficult to implement. It's probably a perception that the people that work on developing them is not the case. So maybe the stakeholder, the operators thinks that it's easier than it is really is. So we perceive the complexity of nature, but yet see that it's really
From a technological point of view. So
yeah.
if we compare other remediation, so we go with agricultural practices in which we are very expert probably, and low cost and easy to be applied. And if we compare with creating wells, injecting compounds and other type of bioremediation, then it require less technology. Maybe it requires more knowledge and more analysis effort because it is a nature-based solution, so it is very sensitive to the environmental parameter and diversity of microorganisms, diversity of the relation plant microorganism and environmental condition and so on. But from a true technological point of view, we are working with so probably this is why it is perceived as a easy to be applied technology.
Yeah, just a small comment related also to what we were talking about before. Since plants tend to accumulate pollutants, probably also need to be considered in the total cost is the cost of disposal of the plants at the end.
Let me say something.
Yes.
So it is true, it is a problem always when we work with plants in polluted soil. What we do with the plants, they are waste, they are special waste, so they need very special care. But before I forgot to comment that I was thinking about food application, because the question was, if from sunflower we obtain oil, can we use it? So my personal view is that we use it, and maybe for human consumption, I would be very cautious, but we can use oil for environmental application. So oil can be used to produce biodiesel, for example. So I see that this could be a potential field of exploration. So it is not my field, but probably oil is food for a non-food application. So it is not going in competition with food. We are working with a very high energy plant, in the case of sunflower. So I think we can explore it is a kind of circularity. So we use a plant to clean that was polluted by energy activity to produce a bioenergy. So even this circularity is a field of interest.
Okay, thank you. And now we move to the main technical strengths of the microbial acid for in-situ bioremediation. What does it mean, numbers? Yeah, we have answered. The highest amount of answer regarding the sustainability using natural and rich indigenous microorganisms. So it is perceived as sustainable. Do you want to comment on this?
Sorry, couldn't follow, so we are commenting this.
Yes.
Okay. Okay. Yes, of course, my comment is that, of course, using enriched indigenous microorganisms, I mean, I can understand this answer because actually with this approach, we are not introducing nothing different from what is already there, at least in terms of microbes. So yes, I can fully understand the reason why this is seen as a strength. I'm curious, but I cannot read actually those.
Targeted nutrient stimulation to boost microbial activity.
Sorry, can
Targeted nutrient stimulation to boost microbial activity and pollutant against removal.
So this is not seen as a strength, and then high adaptability to such specific conditions is also not perceived as a strength. This is probably because, of course, using indigenous microbes with this approach, you are forced to follow, let's say, a preliminary to do a preliminary work in terms of isolation selection from each site that is time and also cost time consuming costly. If you want to do real autonomous fermentation, it is you can adapt, but every time you have work to do before implementing the process. So it's interesting to see this answer.
Okay. Okay, let's move to bias, and we are talking about both, of course, technologies for soil bioremediation, and it looks like the more the one that is more perceived as a barrier is the uncertainty about the performance, followed by regulatory constraints.
I agree. I agree with this result. So the uncertainty about the performance is something that we need to take into account, and I think as more nature-based solution we explore, as less contour we can have. So when we plant the planting, my approach, then the system is self-regulating, and then we do not have the same level of control that we can create in a laboratory reactor. So in a laboratory reactor, we can control all the parameters, so we can probably expect a particular result. If we are going with letting nature, so natural assemblies to do the job in a field, we do not have that control, so it is a balance of aspects and advantages. So I agree that the performance cannot be really always certain.
I want to bring the everyone experience in the remediation context. We have hundreds of sites with our activity for reclamation of soils and groundwater. Is it true that new technologies, bioremediation approaches are not certain, but there is only one technique that is successful with all the context, that is dig up. Dig up was used in the first and the early 2000, but it's not a remediation technique. We transfer the problem from a contaminated site to another. So now it's not acceptable. So we are moving towards a sustainable choice of the technology. Bioremediation is more sustainable. In situ treatment are to be preferred to ex-situ remediation treatment. We must convince authorities that we have to try these new technologies, maybe with pilot tests, as we are trying to do in Ferrara. And one of the most difficult things is to find the equilibrium with all the different authorities that have to approve a remediation project. But this is the new direction that we are taking. No dig and dump, yes, sustainable bioremediation approaches when we can.
Yes, I was just wanting to comment that the fact that the cost is less perceived as a barrier is a strength of the bioremediation.
Just to comment the fact that the two most transparent are in a way linked, because as you mentioned, also the regulatory authorities are worried about the fact that it's a kind of uncertainty in the application of bioremediation. And my other comment is that it's something that we already mentioned before. So the technology could be easy to implement, but the system is very complicated. What we need is something that some tools to be able to monitor the process efficiently. So in this coming year, in the last year, we had the development and the use of omics techniques, we use metabolomics, and many people in the audience use sequencing, other omics techniques that can be helpful just to understand the process going on in the field, particularly in the in situ, but we also have problem X. Not so easy also in the so be one of the future perspectives. So still developing monitoring technique for the processes.
Yes, thank you for this comment. I just would like to add a bit on this, because for sure this was not evident in our presentations, but for example, the microbial modeling activities that we are running with NYMPHE are actually done on these sites, these matrices, these like microbiomes, and will be tested in the last year of the project in these same situations, of course, in the lab, but we will also try to do it in situ. And this is done partially also to address this, let's say, weak point of the in situ bioremediation. So trying to model and to understand in order to adopt or integrate the technology with some solution that can allow to reduce this, let's say, to increase the performance and therefore to reduce the uncertainty in terms of output. So fully understand and agree with this answer. I think the NYMPHE project is already trying to do something also to overcome this aspect using this modeling, which is something we probably a message that we were not able to deliver properly, or we delivered after this questionnaire.
Okay, so let's go to the third section, questions. So how important is the development of innovative bioremediation technology for your organization? Still here we have a distribution of the answers that are towards the fact that the people and the stakeholders, they are they think that they are very important or important. So there are no answer more important or slightly important. So again, the right audience. So here the question was, what is your overall perception of microbial and microbiome-based innovation for environmental bioremediation? So the two most answered points were, I believe, promising. I see strong potential for large-scale application, promising, but still need more real-world evidence of pilots. And then they are the one that the answer is impressive, but limited by technical or regulatory boundaries. So they're optimistic answer. Comment to that?
Very surprised about this. Very surprised about this number. They don't see it as a complex.
No, yes. So thank you for the stakeholders, because this is promising. So that means it's nice. And yeah, it's quite it's rare that they say that it's not complex or uncertain. But it surprised me, because, this is real innovation that is very big on the field. And of course, there is a 40% saying that it's promising, there's still more real work. Totally agree. Because the technology is ongoing, so we still don't know. This is something we have to do in NYMPHE and maybe in other projects. So I totally agree that it's nice that people see it as a positive point. That's it. Obviously.
Is that Celia still connected? Probably.
You hear us?
To comment on this? Maybe not.
Maybe not. So this question is just for perception of microbial and microbiome-based innovation, environmental bioremediation. And no, just looking to comment on that. So maybe, you are very good in convincing people that what you are developing in the lab can be easily applied. So we all know as a researcher that the barrier between the lab and the field is very high, but it seems that the tool are at least very, very promising, so that people do not see this difficulty. So congratulations. That's the kind of problem. Okay. This is a very critical aspect. How do you rate the importance of regulatory and ethical consideration in microbial and microbiome innovations for bioremediation? So this is something related also to genetic modification of microbes and consorts. So the answer is not too many, but most of the people think that the regulatory are very important. So this is very sensitive, particularly in Europe, where we have strict regulation about the use of genetically modified microorganisms in general. So this is something that I would expect in that
or don't want to comment on that?
Yeah, yes, sure.
Yeah, but this is totally expected. It needs from the beginning, we, in particular, you can see that GMO, GMO is something super terrible. That's why I'm particularly saying we believe that areas will not have GMO, will have evolved microbials. But it's true that at the end, ethical and regulatory considerations will be super important. Because probably by law, we will not be able to use our technology in the field. And this is something that as researchers, we have to really happy because, I also try not to say inflation. I'm going to think that, people think about methods are mutations like bad things. I mean, in the collective mind, immediately is Godzilla, kind of wood or Resident Evil is the bad things. And in nature, it's not true. Mutations are usually neutral, not functional or detrimental, they are bad for the organism. And sometimes they are good. We all are mutants. We are all evolution. And this is something that we have to really explain in university and schools and everywhere to make people understand that evolution is nice, it's good, it's a source not problems, but solutions. This is my, we have to transfer the most difficult line, which is the regulatory models. But this is a thing that in Europe we really have to work.
Another role for the university, and my question to you is do you think that is in these times of social sharing of news and fake news, this perception, this bias perception increased in the last 5 years?
I don't really know. We should check the surveys and something, but still I think that people is strongly against it. I mean, I think it didn't change. It is something we have to change at all levels. I don't know if people, but yeah, people is changing their mind, and then you have to comment if you want it. I don't know. There's a pity that this thing is happening because it happens. The possibility of flying technology, and we need now the doing clearly. Yeah. But yeah, as a researcher, we have, some other stakeholders has to move forward.
Was the last one?
What was the last question? Do you want to close the session?
Yeah, sure.
Yes.
Oh, sure. Yes.
Thank you very much. I will try to do so because I know I'll go for that, but I wish to thank you all NYMPHE for considering RemTech to present your work. The RemTech is quite important, okay? Because on this room is actually where data is collected, information is generated, science progresses, and downstairs it's where all these results eventually they will find a way, they will commercialize, and they will be practiced by economists. Okay? People like us. Some of us we study in the university, become researchers, some of the other people they become consultants, and some of the other people, of course, they will fill the positions of the regulatory authorities. So pretty much we are there. It happens to be that I am also a researcher, but I'm also working as a consultant. So I'll try to put all these things into context. You mentioned about sustainability. That's super important. One of the things that you may consider in the future when you try to scale up these things is after you perform all these remedial methods, there might be bounce back effects. So what it happens if you vegetate the area, then you crop all these things. What is the validation? What is the remediation results? Are they still remain there or they bounce back? And that will create trust. If you can, have the results that everything is working, that's sustainability based. At some point, you mentioned financial costs and OpEx, and you mentioned that this is quite low, which is great, super great. I wouldn't worry about it, frankly. Because if you put everything into the equation with whatever we consider as remediation methods, they are super expensive, and most of the time we don't consider the social costs. If you put the social costs into the equation, that's fantastic. People that want this kind of methods. Okay, some of the contractors, when they want quick remediated method, contaminated land, because they have, of course, to develop it, invest it, and make quick money. But in some cases, that might not be the thing, especially when you have large areas that they have to remediate it over time. And doing the quick way, it might not be the right way. And considering the social acceptance, this most probably are the methods that in the future will have to be implemented anyway. Training. Training is quite important. You mentioned here that most of the training should actually happen at the consultancy level. I agree, but also the seats should actually be implemented during the university. Why? Because in that case, we have established the communication both ways. Okay, so the people that are in the university, they can communicate with the practitioners and the regulators, so it becomes an ecosystem. So I don't think that we should isolate ourselves. We should communicate more and more, and actually even, you may consider, of course, master's degrees, but this should also start at the bachelor level, this is very important. When it comes to soil, the ecosystem services is very important. We need to preserve this. I will also think this. We mentioned also regulatory constraints. I would also worry about this because, again, regulators, they tend also to learn from us. We bring solutions. Okay, that this is prohibitive. Okay, but if we demonstrate that it works, also the policy makers these days are well-educated scientists, and they take all these things into consideration. One of the most important things, and this comes from the social science perspective, it doesn't matter how good your data is, but it matters who the communicator is. Who is the one that brings trust between the public and the science? It's the middleman. That's something very important because most of the time there's a good research being produced, and sometimes it's perceived badly by the public. And this is one of the things that you mentioned about the net education. It's about educating the public, making sure that they understand, and making sure the ones that they really transfer the message are trustable. Sometimes it happens by the wrong people, and I think this is super important. And I will close with this. Of course, we have moved from dig and tap back in 2000. Yeah, it was one of the ways, and we were still learning. Of course, at the time, things were far more strict, and we had soil guideline values, we had all sorts of different systems. Now, most of the regulatory authorities and environment agencies, permitting authorities, in Europe at least, they have adopted a risk approach method. So when actually you offer what's going on and what is the risk perceived, sometimes you don't have to go to extremes. And if you basically you can offer and demonstrate that this kind of methods, they have everything in balance, okay? But if there is within an acceptable risk, combined with social costs and financial costs, usually the permits are not the problem. So, thank you very much.
So, thank you really very much for your interesting and useful
comments and suggestions. So with this, I think we can close this workshop. Before doing it, first of all, let me thank all the participants that were present today here in the room and online. Thank you very much, and also thank you for participating to the debate and for letting us know through the questionnaires what you think about what we were speaking about today, because this is really valuable for us. So the first thing is to you. And then, of course, I would like to thank Ina and Mauro for organizing and hosting this very nice event here at Remtech. Of course, Remtech for hosting with this audience and with our colleagues from Innovo who are taking care about the organization, let's say, from the technical part and the questionnaires, and of course, last but not least, all our colleagues in NYMPHE who were here, and unfortunately, Silvia, I'm sorry, I know the reason online, so all of you for your contribution to this work. So thank you very much, and hope to see with you and discuss with you again later today or in future occasions.
Thank you. Thank you all. And for the people that are still online, they have closed in the room. We have plenty of other sessions in the afternoon. We have PFAS management strategies. We have the live demo. We have the PFAS in groundwater, bioremediation strategies. So there is plenty of other sessions that, if you are interested, are all free of charge, and you can follow it online. So thank you again, and see you soon.
See you soon.